No, thank you so much for the invitation to talk. It's a really great talk by Lucia as well, and she obviously introduced the topic of dormancy really well in breast cancer. And today I'm going to be talking about how the role of aging can actually promote reactivation from metastatic melanoma dormancy. And it's going to raise some issues as regards to how conserved some of these dormancy programs are between things like melanoma, breast cancer, prostate cancer, so on and so forth. So before I start, I thought I would quickly plug the lab. So I actually start in May, so we haven't fully opened our doors yet, but we are doing a lot of cool techniques, and hopefully I can show you some of them today. So if you are interested, send an email to mitch.fane@gmail.com. Our current lab member is Hank Fane. He is my wiener dog. Unfortunately, he's about to submit his thesis, as you can see here. So he's going to be leaving soon. So I'm definitely going to need to start filling the lab up. So yeah, if you're interested, please send me an email. So today I'm going to be talking about melanoma. So just a brief introduction into melanoma. It is the deadliest form of skin cancer, and it's due to the malignant transformation of melanocytes located within the epidermis of the skin. So what we see as this melanoma continues to progress towards different stages of metastatic disease is that it grows deeper and deeper into the dermal tissue, and then eventually it undergoes this concept known as phenotype switching, which I'll discuss a bit further. But basically, it starts to become less proliferative, and it gains invasive or motile capabilities. And eventually, it can start moving away from the dermis and invading deeper and deeper until it can interact with the vasculature or the lymphatic system, where it can start to spread to distant metastatic organs. The lymph nodes are a stage 3, and it can spread to other distant organs in stage 4. Now, when we look at some of the problems when it comes to treating melanoma, one of the major problems is that melanoma can metastasize to most organs within the body. So we see that melanoma mainly metastasizes to the lung, the liver, the colon, and the lymph nodes. But indeed, there are many different case studies that show that it can almost grow in all organs within the body, depending upon specific contexts. So what's really interesting is that there's been a revolution in the way that we treat melanoma over the past few decades with the advent of immunotherapy. So PD-1 and CTLA-4 have come in and done a really good job in a number of different patients. And even recently, just a few years ago, the FDA also approved anti-LAG-3 as another checkpoint inhibitor that can be used as well. The problem is, though, that not all patients respond to or are eligible for therapy, unfortunately. And even the patients who do respond to often show very remarkable responses initially. There's a large majority of them that will ultimately relapse and become resistant to these specific therapies. And then this metastatic event starts to become much worse. And now they don't have any treatment modalities to basically keep that in check. So one thing we really have to understand better to increase durable therapy is this metastatic cascade and how we can efficiently target cells, not just at a primary tumor, but at all of these distant metastatic sites. So another thing that's not often probably thought of in melanoma, and indeed many different cancers, is that they're often referred to as a disease of aging. Most people think of skin cancer as mostly a sun cancer, staying out in the sun too long, but indeed the median age of diagnosis of melanoma is 63 years old. And you can see that the incidence of most cancers, but particularly melanoma, increases drastically with age, as you can see here. We also know that prognosis is worse for the elderly, and also therapy is not as effective in older patients. And what's really interesting is things like the World Health Organization have suggested that with advancements in technology and healthcare, we're going to drastically increase the life expectancy of people. And while this may sound like a great thing, one of the kind of major issues it's going to do is increase the incidence of many of these age-related cancers like melanoma. So we really have to understand how aging can intersect with cancers such as melanoma, and indeed how it intersects within that metastatic cascade, so we can start to create these durable therapies, because it's just going to become a much worse problem as we increase this aging population. So indeed, when we look at how we age as healthy individuals versus aging within a tumor, we see really drastic changes within the microenvironment of our tissues. So this is a review that Ashi and I wrote in 2019, basically looking at the differences between a young and aged healthy and tumor microenvironment. And we see a large number of different stromal cells change during this aging process. So one of the major components that changes over time are fibroblasts. So fibroblasts are responsible for secreting soluble proteins and also depositing extracellular matrix, which makes up the structure of almost all of our tissues. And it's one of the key components or key stromal components of almost all tissues within the body. We also see a really dramatic change in angiogenesis, where usually over time as we age, we see a decrease in angiogenesis within the body. However, within these tumors, we tend to see, in most cases, an increase in angiogenesis. And of course, we also get a really dramatic difference in immune cell infiltration based on the different subtypes that can come in to different tissues and also into these aged tumors versus young tumors. And these can have really dramatic effects on how aggressive these tumors grow and also how aggressive these tumors can disseminate and metastasize to different organs. So when I first joined the lab, my question was to focus specifically on metastatic tissues. A lot of the studies that had been done in our lab and a lot of other labs had focused on the primary tumor. And I wanted to look at how metastatic tissues change with age and how this can modulate the metastatic cascade. So I first started looking specifically at the lung, and what we used is this Yum 1.7 syngeneic melanoma mouse model, which was created by Marcus Bosenberg in Yale. And what's really unique about this mouse model is that we can use the exact same genetic tumors and we can put them into the exact same genetic mouse model and form either primary tumors or lung metastases via tail vein, which Lucia talked about before. And the only difference is that we can do this into younger mice and aged mice. So we consider our younger mice in our study to be 8 weeks of age, and we consider our aged mice to be 52 weeks of age. So what's also interesting is the geriatric age of mice is usually around the 18 to 24-month-old range. One of the issues with that is it takes a very long time to age them to this point, and it is very expensive. So we began simply by looking at the differences between this 8-week and 52-week-old mouse. So the 52-week-old mouse is usually relative to a middle-aged human, maybe in the 40s to 50s, but we indeed did see very dramatic differences at this point. So what we found is that if we form a primary tumor within these specific mice, we see that they grow very differently in the young versus the aged skin. So this is a primary tumor growth model where we measure the primary tumor at different time points throughout tumor progression, where once they reach 1,500 millimeters cubed, we have to euthanize them due to an ethical tumor burden. And what we find is that the young mouse skin is able to grow the primary tumor much more efficiently compared to the aged mouse skin. However, if we move to these tail vein models where we inject the cells directly into the tail vein and they go directly into the lung, we see the exact opposite effect in which melanoma cells grow much more efficiently in the aged mouse lung relative to the young mouse lung. So indeed, it seems that these organs show this age specificity in how they regulate melanoma growth. And I wanted to determine why there was this age specificity and how it actually contributed to the metastatic cascade of things like dissemination versus outgrowth at these distant metastatic sites. So to first look at this, what I did is I formed primary tumors in our young versus aged mice using our Yum 1.7 model, but this time containing an M-cherry fluorophore. And it basically allows us to look at how these cells disseminate into the lungs by performing IHC at different time points throughout tumor progression. So 3 weeks was the first time in which we were able to see these cells successfully seed within the young versus the aged lung. So you can see when we stain for M-cherry, these red cells here are the M-cherry positive melanoma cells. You see that they seed as single cells in both instances. And when we looked at the number of cells that were able to successfully seed, there was no real difference in the number of cells that reached at this specific time point. However, if we look at week 5, which is around the time in which these mice reach their ethical tumor burden, we start to see really dramatic differences. So we see in the young lung that they maintain this single cell state. Potentially, they're suggesting that they're not able to grow out and they're undergoing this prolonged period of dormancy. Whereas in the aged mouse lung, we're starting to see much larger metastatic tumors growing, suggesting that the aged mouse lung is inducing a more proliferative phenotype. So to investigate this further, what we did is we got our single M-cherry positive cells in the young lung and co-stained them with Ki-67, which is a marker for proliferation. And you see that they stain negative for Ki-67, indeed suggesting that they've exited the cell cycle. Whereas if we look at the aged lung, even if we find a very small metastatic tumor, we see that it stains very positive for Ki-67. So that was in our subcutaneous model. So if we move to our tail vein model, indeed, we can show that if we inject the same number of melanoma cells into the young versus the aged mice, we see that they grow out much more efficiently in the aged mouse, as indicative of these deep purple regions, whereas in the young mouse, they're able to start growing out, as you can see by these purple regions here, but they're not able to grow out as efficiently as the aged lung. And if we stain these metastases for Ki-67, we get this really dramatic difference where they stain much more positive in the aged mouse lung compared to the young, indeed suggesting that the aged mouse lung is inducing more of a proliferative phenotype. So then this begs the question, what is actually going on? And Lucia did a really good job of introducing single tumor dormancy. So I'm not going to talk too much about it. But in our context, given that dormancy can happen via a number of different mechanisms, what we're seeing is this kind of intrinsic growth arrest of tumor cells towards this quiescent state, which is a reversible exit from the cell cycle. And what's really interesting is a lot of the time, there's these really great models of dormancy in breast and prostate cancer, but there hasn't really been any good models of dormancy in melanoma. But it's actually been shown via a number of different case studies that this dormant tumor cell effect does occur in melanoma. And this actually comes from organ transplantation studies. And what's been found is that these patients in these different case studies have donated either their kidneys or their lungs, and they weren't aware that they had these dormant melanoma tumor cells present within these organs. They either never had melanoma diagnosed in their life, or they had a primary tumor that might have been removed very early on in life and they were considered to be disease-free. So we don't have any really good clinical markers for tumor dormancy in melanoma, so you can't screen these organs as you're putting them into these patients. So these patients receive these organs unaware that they have these dormant tumor cells, and then eventually over time, they get these very large metastases that occur. And when these metastases were analyzed, it was actually found that they were from the original donor and not from the patient who received these organs. So these really interesting case studies beg a few different questions in terms of how dormancy can act within melanoma specifically. So the first one is that some of these case studies took decades. One of these lung case studies took 31 years before these cells were able to reactivate, potentially suggesting that this tissue has to age to actually allow these cells to reactivate. These patients are also on immunosuppressive therapies, potentially suggesting that the immune system might play a key role in maintaining tumor cell dormancy. And of course, these patients don't have any primary disease. And interestingly, even the patients who donated their organs didn't have any diagnosable primary disease. So this shows that this process can occur irrespective of any primary disease being present for melanoma. So when we talk about this idea of these cells switching between a dormant state and a reactivated proliferative state, it begs the question, how are they actually doing this? And one of the key models specifically in melanoma is what's referred to as the phenotype switching model. And what this model argues is that melanoma cells are able to interact with extrinsic cues within the microenvironment, be it in the primary tumor or within metastatic tissues. And based on these extrinsic cues, they're able to adopt a large number of different phenotypes. So the two main phenotypes that are often characterized in melanoma are this proliferative phenotype, characterized by the MITF transcription factor and also the canonical beta-catenin pathway, versus this very slow cycling but highly invasive melanoma phenotype. And we've shown previously that it's been characterized by expression of Wnt5a and also by expression of the tyrosine kinase receptor AXL. So given that we were seeing this dormant phenotype, I wanted to ask, where does dormancy fit within this model of progression? So what's really interesting is if you look at the melanoma, the metastatic cascade of melanoma, what we usually find is that you get a malignant transformation of a melanocyte, you get a much smaller, slower growing tumor, and eventually it gains the capacity to grow out of control. Eventually, as these tumors continue to grow, you start to get this phenotype switch towards an invasive cell state based on changes within the microenvironment. And then once this phenotype switch occurs, they start to disseminate into the lung. So one of the questions that I had coming into this study was that Wnt5a drives this very slow cycling phenotype within our phenotypic switching model. So I begged the question, once they get to the lung, are they maintaining Wnt5a to induce this dormant state in the young lung? And in the aged lung, are they actually reducing the level of Wnt5a expression to allow them to reactivate? So what was really interesting is while I was starting this project, these papers came out in prostate cancer showing that both the axial tyrosine kinase receptor and Wnt5a was able to induce and maintain prostate cancer cell dormancy within these bone metastatic niches. So it begged the question, can these pathways be conserved between things like prostate cancer and melanoma to drive a dormant phenotype? So to investigate this, I first specifically looked at Wnt5a. So we went back to our mouse model. So in our young mouse model, we have these single tumor cells present. As you can see here, that we have a cluster of single cells that are Ki67 negative. And when we stain for Wnt5a, you can see that they stain very positive for Wnt5a expression. However, if we get a much larger melanoma metastasis that we can see is very positive for Ki67, if we stain for Wnt5a expression, you can see that it doesn't express high levels of Wnt5a. So this indeed suggests that it seems that Wnt5a is only expressed in these Ki67 negative single cells and not in these larger proliferative tumors in the metastatic lung. So next we wanted to determine if we manipulate Wnt5a levels, are we able to change the metastatic cascade? So what we did is we got our young 1.7 cells and we put this tetracycline inducible SH Wnt5a construct in it and formed primary tumors in our young mice. And how this construct works is that we treat with doxycycline, it induces this TEP promoter to basically induce expression of this short hairpin Wnt5a, and it knocks down Wnt5a expression, as you can see here. And when we knock down Wnt5a, you can see we get a decrease in some of these dormancy markers like AXL and P21, and an increase in the proliferative marker MITF. So we next wanted to determine whether knocking down Wnt5a was able to regulate tumor growth in vitro and within a primary tumor model. And indeed, when we knocked down Wnt5a by treating with doxycycline, we got an increase in tumor melanoma growth in vitro, and we also got an increase in primary tumor growth in vivo. So this would suggest to us that Wnt5a is responsible for driving this slower cycling cell state. So next, we wanted to determine what role Wnt5a plays in dissemination and outgrowth within metastatic tissues. So the first question I wanted to ask is, what happens when we knock down Wnt5a within the primary tumor during a very early time point in tumor development? So what we would do is we would form primary tumors in these mice, and we will allow these primary tumors to grow for 3 days. And then we would start treating with doxycycline. So basically during this entire time of tumor development, they have this knockdown Wnt5a expression. And we treat these mice with doxycycline by putting it in their water. And we allow them to have this for 35 days. So we get a full 35-day primary tumor growth. And then after that, we take the lungs and look for metastasis. And what we find is that in our young lung, usually after 35 days, we're only able to see these single cells, as you can see here. But when we look at the lung, if we start treating with doxycycline very early on, we get a significant decrease in the number of cells that are able to reach the lung at this specific time point, indeed suggesting that Wnt5a is involved in this initial dissemination step. But the question that I had is, what happens if we knock down Wnt5a in cancer cells that have already reached the lung? So to answer this question, I actually started treating a doxycycline at a much later time point. So I waited for 3 weeks, because if you can remember back to my first slide, I actually showed that it took 3 weeks for these single cells to seed and become viable within the lung. So I allow them to seed at 3 weeks, and I treat with doxycycline to then knock down Wnt5a expression within the melanoma cells in the lung. And indeed, if we do that, we start to see much larger metastases forming at this 5-week mark compared to the no-dox control. So based on this, it seemed that Wnt5a needed to be knocked down in metastatic tumors to allow efficient outgrowth. So we then switched to our tail vein model to look at this at a more macro metastatic level. So as I mentioned before, when we inject in the tail vein, it can take up to minutes for the cells to reach the actual lung and start seeding. So we allowed a 3-day period for these cells to seed within the lung, and then we started treating with doxycycline. And when we do this, we find that if we knock down Wnt5a after cells have successfully seeded in the lung, we start to get really large metastatic tumors compared to our no-dox control that only had these small tumors located here, as you can see. However, if we stain for Ki67, we also find that in these larger Wnt5a negative cells, they're actually able to induce much more Ki67 expression relative to the no-dox control. So the final experiment we did is the actual opposite of this. This time we moved to aged mice. And we see after 35 days that the aged mice have much larger metastases relative to the younger mice. And what we did this time, however, is we overexpressed Wnt5a instead of knocking it down. And when we overexpress Wnt5a, either at day 3 or day 21, what we find is that we're able to get a large number of cells reaching the lung because it's driving this dissemination effect, but they're not able to grow out as these larger metastases. They're only able to form these single cells, as you can see here. So indeed, this seems to suggest that Wnt5a drives this dormancy phenotype initially once it gets to the lung. And what we find is that in the young lung, it seems to maintain this high level of Wnt5a expression to maintain dormancy, whereas in the aged lung, it seems to be able to knock down Wnt5a to induce reactivation. So the next question I wanted to answer is what was happening in the young versus the aged lung microenvironment to alter these genetic programs to manipulate dormancy and reactivation phenotypes. So a lot of work that was done previously in our lab when I joined was focused on how young versus aged dermal skin fibroblasts were able to alter the secreted soluble proteins and how they were able to basically regulate immune cell signaling, melanoma cell signaling, angiogenesis, and all of these other different stromal components within the primary tumor. So it kind of begged the question for me, given we were seeing this dramatic difference in the lung, are aged lung fibroblasts potentially secreting different soluble proteins relative to younger lung fibroblasts? And are they potentially inducing this reactivation from dormancy? So to investigate this, what we did is we sourced healthy human young and aged lung and skin fibroblasts from healthy human patients. And we created this 3D sandwich reconstruct where basically we would put our fibroblasts of interest into a top and bottom layer, and we would have GFP melanoma cells located within the center. And we could use a microscope to look at how these cells proliferate over a 4 to 5 day period. And what we found, which phenocopied what we saw in vivo, is that the melanoma cells were able to grow much more efficiently with aged lung fibroblasts relative to young lung fibroblasts. However, if we did it with skin fibroblasts, it was the exact opposite effect, in which they grew much more efficiently with young skin fibroblasts relative to aged skin fibroblasts. So I chose to specifically focus on how aged versus young lung fibroblasts were regulating tumor growth and inducing our metastatic phenotype that I discussed previously. So to look at this, I then took conditioned media from young versus aged lung fibroblasts to see if it was the soluble proteins being secreted that induced this effect. And indeed, if I treat melanoma cells over a 10-day period, eventually they start to grow much more efficiently in aged lung fibroblast conditioned media relative to young lung fibroblast conditioned media. We next then treated with 3 different young versus aged healthy human lung fibroblast conditioned media in these melanoma cells and looked at protein expression. And you can see if we treat with aged lung fibroblast conditioned media, we get a dramatic decrease in a lot of these different dormancy markers, particularly Wnt5a, which is this top band up here. You can see it completely disappears, and also AXL as well. And we also get an increase in these proliferative markers. So based on this, it seemed that soluble factors were being secreted differentially between young versus aged lung fibroblasts. So I wanted to investigate what were the actual factors being secreted differentially. So I did proteomic analysis with the Wistar Institute, where we basically looked at the factors that were being increased by young lung fibroblasts, which are seen here on the left, versus aged lung fibroblasts, which are seen here on the right. And one thing that stood out to us almost immediately was this SFRP1 factor. And the reason why it stood out to us is that a lot of literature had shown that SFRP1 actually inhibits Wnt5a signaling in a number of different pathological models, but it hadn't been investigated within melanoma. So my hypothesis was that aged lung fibroblasts started to secrete high levels of SFRP1, which knock down Wnt5a and induces this phenotypic switch from this dormant state towards this proliferative state in the lung and allows these cells to grow really efficiently in the aged lung. So to investigate this in vitro, what we first did is simply treat melanoma cells with recombinant SFRP1. And indeed, what we found is that we get a decrease in Wnt5a expression and an increase in the proliferative marker MITF. And we also get an increase in melanoma growth over 10 days when we treat with SFRP1 relative to a PBS control. So the final experiment we did is we wanted to determine whether or not SFRP1 was inducing reactivation from dormancy and allowing these cells to grow out efficiently in the aged mouse lung. So we formed a primary tumor in our aged mice, and we allowed this primary tumor to grow for 3 weeks to allow that initial dissemination to occur in the lung. We then started treating with a neutralizing antibody against SFRP1 versus an IgG control. And then over this 2-week period, we finally looked at what happens to the M-cherry metastases after 5 weeks. And this SFRP1 antibody basically depletes levels of SFRP1 within the mouse. And what we found is that in our IgG aged control, as we usually see, we get much larger metastases forming. Whereas when we neutralize SFRP1 and deplete its levels in the mouse, we're only able to see these single cells forming in the majority of these mouse lungs now. So in summary, hopefully what I've been able to show is that aging promotes this reactivation from metastatic melanoma dormancy relative to the young lung. We see that aged lung fibroblasts over time eventually start to secrete high levels of SFRP1, and they're able to successfully downregulate these dormancy programs like Wnt5a to promote reactivation from dormancy and allow melanoma growth and metastatic progression. And if there's one key thing you can take from this talk, it's the fact that there is no one gene fits a metastatic model. Previously, a lot of people say, Wnt5a drives metastasis. It was the hypothesis of our lab. But as you can see, we show that it has to have a really fluid expression level during different time points throughout progression to really allow efficient metastatic progression, such that it has to be high within these primary tumors to allow dissemination, and then it has to be reduced to allow reactivation. And I'm sure when it has these insults like therapy, or if it's being attacked by an immune cell, Wnt5a is going to have to change as well to basically allow it to survive and be able to maintain these metastases. So yeah, that's kind of one key message to take away from this, as well as aging is extremely important when it comes to modeling these age-related diseases like cancer. So the future challenges and questions that I have for modeling dormancy, specifically in the lab that we're going to be opening in May, is how do changes in other stromal components in the lung microenvironment promote reactivation? So we have 2 key projects that we're very interested in right now. So the first is the role of the aging in ECM-induced reactivation. So how does ECM remodeling change with regards to age? We're very interested in the lung immune microenvironment. We have some very cool data at the moment, which I hope to share in the future once we're able to analyze all of that. And then, of course, how does aging affect other target organs? And we've recently learned of a technique where we can start to form really nice liver metastases. And we want to investigate the difference between young versus aged liver metastases relative to the lung to see if there's different potential dormancy and reactivation programs going on and different stromal changes going on between these different organs. Because if there is, then that poses a real threat in how we're able to treat these different metastatic tumors based on their specific site. So with that, just a huge thank you to everyone in my lab. Of course, Ashani Wirana has been the most amazing mentor that you could ask for. I've been able to work with a lot of really great people over these years to produce this project and a lot of other things. And yeah, we just have a really great lab, and it's such a great time being there. So I'm hoping that when I build my lab out, I can build it 1/10 as good as what Ashi has been in terms of the culture and the environment that we have there. So yeah, thanks for that, and happy to answer any questions.
Thank you very much. That was really interesting. So now it's time for questions. You can raise your hand, or you can also write them down and I can read them out for you. So I had a question because you mentioned that, of course, some melanoma patients are treated with different drugs like immunotherapy and so on. Is there something known about the impact of immunotherapeutics on dormancy of cancer cells?
That's a really good question. It's something we're very interested in doing, basically treating these mice with immunotherapy and seeing if we actually alter the effect of dormancy. There isn't a huge amount known yet, just because there's so few modeling systems being able to model dormancy of melanoma. So, I mean, yeah, it's definitely something that we're very interested in. But I think some of the key data that we have that will be coming out soon is that the immune system is playing a really key role in maintaining this dormant state. So I think that hopefully these immunotherapeutics will be able to either maintain them in this dormant state or allow these cells to be eliminated while in this dormant state.
I see. And do you also think that maybe some tissues age differently than others or faster, and they might be more prone to develop metastasis?
Yeah, of course. Yeah, definitely. That's why we're trying to switch to a liver metastatic model to see if there's differences between the liver and the lung. Colon is also very interesting, probably a bit harder to model compared to the other two, but we'll definitely move to liver. And the whole point of doing the liver is to just do a really kind of strict comparative analysis between the skin primary tumors versus the lung versus the liver, and seeing, you know, what happens with regards to age and how these cells are able to grow out differently, and the different immune responses, and of course the different ECM, how that affects tumor outgrowth. Because there is a lot of literature evidence, and I've talked to a lot of clinicians that say that you get these really site-specific metastases depending upon the patient. So some patients will only have lung metastases, other patients will have metastases everywhere but the lung, and yeah, so it seems to be depending upon the patient and obviously the heterogeneity, it seems to really dictate where these melanoma cells go with regards to kind of seeding. And yeah, that's a really good question that I hope to be able to investigate in the future.
And then probably there is a question from the chat. They say, great talk. Do you think regenerative capacity of the liver tissue might impact dormancy in liver metastasis?
Yeah, I mean, that is a really, really great question. I honestly have no idea. I think honestly, I have no idea. I haven't really looked into that point enough, but it's really something to consider, and I'm sure it would have a major impact, and probably is one of the reasons why we get some cells able to, you know, be able to form these efficient mets in the liver where other cells can't, and they form better in the lung. So I'm sure it depends on the specific melanoma type once it does disseminate, and you know, mutations, all of those types of things. I'm sure they play a key role in how they're able to go into these tissues, remodel these tissues. And yeah, even with the aging, I think over time we would probably start to lose a bit of that regenerative capacity in the liver. And I think with that, it might actually open the door to having much more efficient metastasis into the liver. So that's a key question that we could try and ask as we're doing our young versus aged liver studies.
Hopefully we see the results coming out soon.
Fingers crossed.
Fingers crossed. Thank you very much. I think now there are no further questions, but I guess if anyone has a question, they can reach you, Twitter, email, or anything.
Mm-hmm.
Thank you very much, both to you and Lucy. I hope you had a nice time, and I wish you all a nice day. See you soon.
Yeah. Thanks for the opportunity. Thank you, everyone, for coming as well.
Bye.